You ever stop and think about one of the biggest, most stubborn mysteries in all of medicine? It's a question that really gets at the limits of our body's own amazing repair manual. It seems like a whole chapter has just been left blank. So let's dive in and investigate why.
I mean, think about it. Our bodies are incredible healing machines. We see it all the time. But when it comes to the central nervous system, the brain and the spinal cord, that amazing ability to heal just disappears. So what is it? What makes these critical nerve pathways so fragile? That's the question right at the heart of this explainer.
Okay, so to even begin to get our heads around this, we have to talk about a huge paradox that's happening inside our own bodies. It's a contradiction that has really puzzled scientists for decades, and it sets the stage for everything we're about to uncover.
See, our nervous system is basically split into two big teams. On one hand, you've got the peripheral nervous system, or PNS. These are the nerves running through your arms, your legs, all your limbs. If you cut a nerve here, it has this incredible ability to regrow. But on the other hand, you've got the central nervous system, the CNS, the big command center in our brain and spinal cord. And after an injury here, nothing. The axons, those long communication cables of our neurons, just fail to regenerate. And that right there, that's the heart of the whole mystery.
So what's the deal? For a really long time, everyone just assumes that CNS neurons were fundamentally broken, that they just couldn't regrow. But then, this landmark study back in 1981 completely flipped that idea on its head. Scientists took a piece of a peripheral nerve, a place where growth is welcome, and they grafted it into an injured spinal cord. And you know what happened? The CNS axons actually started to grow right into it. This was the first giant clue. The neuron itself isn't broken. The problem wasn't the neuron at all. It was the neighborhood.
All right, so if the neuron can grow but doesn't, what's stopping it? What happens at that moment of injury in the CNS that just stops it dead in its tracks? Let's put on our detective hats and zoom in on the crime scene.
Okay, quick refresher, just so we're all on the same page. The axon is the neuron's main communication wire. It's the part that gets severed in an injury, and it's the part we desperately need to regrow. And when it is growing properly, its tip is this dynamic, amazing structure. And that structure is called a growth cone. Now, when we're developing or in a part of the nervous system that can regenerate, this growth cone acts like a tiny molecular hand. It's constantly feeling its way through the tissue, sniffing out chemical cues, and pulling the axon along to where it needs to go. It is an absolute engine of exploration and connection.
But in an adult CNS injury, something goes horribly, horribly wrong. Instead of forming that beautiful, dynamic growth cone, the severed axon tip just swells up. It becomes this disorganized, sealed-off mess called a retraction bulb. It's the physical embodiment of failure to grow. The engine is stalled. A wall's gone up. The question is, why? What's causing this total shutdown?
So following that first big clue from the 1981 study, researchers started looking at the environment, and boy, were they onto something. It turns out that the scene of a CNS injury becomes a seriously hostile environment for any axon trying to grow.
And this is where you really see the difference. In the PNS, you get these helpful cells called Schwann cells. They rush in, they clean up all the debris, and they form these perfect little guide rails for the new axon to follow. They're the support crew. But in the CNS, you get the exact opposite. A dense, tangled web of cells and proteins called a glial scar forms. Now, its job is to quarantine the damage, which is good, but in doing so, it creates this massive physical and chemical barrier. It's not a cleanup crew, it's more like biological crime scene tape just walling off the whole area.
And yeah, this isn't just a physical wall. It's packed with chemical do not enter signs. Over the years, we've identified the main molecular suspects here. Proteins like NOGO and MAG, and these molecules called CSPGs. They're all over the scar, and they basically act like little guards, shouting, "Stop, go back!" They actively repel that growth cone and cause it to collapse into that dead-end retraction bulb.
So, for a long, long time, scientists thought that was it. Case closed. The bad guy was the hostile environment. But it turns out there's a huge plot twist. The environment isn't the only problem. The neuron itself is in on it. It's an inside job. So get this. As our central nervous system finishes developing and matures, our neurons go through this major change. To create the stable, precise wiring that we need to function, they actively shut down the very genetic programs that let them grow in the first place. For most of our life, it's a feature, not a bug. That is, until you get an injury. Then, it's a catastrophic bug.
And this whole on-off switch is controlled by these master genes. Take the KLF family of proteins, for example. They're perfect examples of these switches. As our neurons mature, the levels of some of them, like KLF4 and KLF9, go way up. These act as powerful brakes on growth. And at the same time, the levels of their pro-growth cousins, KLF6 and KLF7, go way down. The neuron literally puts its own foot on the brake and takes it off the accelerator.
And there's another major player on the inside, a pathway involving two molecules, PTN and MTR. The easiest way to think about it is this. MTR is like the gas pedal for cell growth. When it's on, the cell builds things. And PTN, that's the emergency brake, hardwired to MTR. In our mature CNS neurons, that PTN brake is slammed on, keeping the powerful MTR growth engine completely shut down. Okay, so now we've got our suspects lined up. We've got the hostile environment outside and these powerful brakes on the inside. Armed with this knowledge, scientists aren't just staring at the problem anymore. They're figuring out how to crack the case and cook up some pretty clever solutions.
Now remember how those peripheral nerves can heal? Well, scientists discovered something wild. If they took a sensory neuron and just injured its peripheral branch, the part in the arm say, it sent this powerful wake-up call all the way back to the main cell body. This conditioning lesion, as they called it, actually flipped all those dormant growth programs back on. This was huge. It was the proof of concept everyone was looking for. It showed that these internal brakes can be released. The trick is just figuring out how to do it on purpose.
And this discovery basically laid out the modern game plan for repair. It's not about one magic bullet, it's a full-on, multi-pronged attack. Step 1. Clear the path. You use special enzymes to basically eat away at that glial scar. Step 2. Release the brakes. You go inside the neuron and you shut down molecules like PTEN, which takes the foot off the brake and reactivates that mTOR growth engine. And finally, step 3. Build a bridge. You can graft supportive cells to create a friendly pathway to guide those newly awakened axons right across the injury site.
You see, by hitting both the outside problems and the inside problems at the same time, researchers are finally starting to see real significant axon regeneration in animal models, the kind of stuff that was considered science fiction not too long ago. So what does the future of all this look like?
Well, the path forward is complex, but the goals are becoming clearer every day. It's about combining these therapies, because we know there's no single magic bullet. And it's not enough to just get axons to grow. You have to make sure they connect to the right targets. That means we also have to use things like rehabilitation to strengthen those new, useful connections. And the ultimate goal? It's to learn the full genetic language of the neuron so well that we can essentially reprogram it, turn it from a static, mature cell back into a young, growing one, ready to repair itself.
So it started out as this massive paradox. A body that can fix almost anything except its most important wires is slowly but surely becoming a solvable puzzle. We've found the culprits outside, we've found the switches inside, and we're learning how to flip them. We are learning to speak the language of neuronal repair, and that leaves us with one final, and I think very hopeful question. The issue is no longer if we can speak it, but how fluently.